Assalamualaikum warahmatullahi wabarakatuh and good day. Today we are going to continue our lecture session. Now we continue to metabolic pathway. Usually, metabolism is divided into two categories. First is the catabolism, second is the anabolism. Catabolism, which yield energy, an example being the breakdown of the food in a cellular respiration, while the anabolism use energy to construct components of cell such as organic compounds, for example protein and nucleic acid. For catabolism is a set of metabolic process that release energy. This include breakdown and oxidizing food molecules. The purpose of this catabolic reaction is to provide the energy and components needed by anabolic reaction. Anabolism is a set of constructive metabolic process where the energy from the sun released by catabolism is used to synthesize the complex molecule. In general, the complex molecule that make up cellular structure are constructed step by step from small and simple precursor. Now we move to the enzyme structure and function. Enzymes are catalytic molecules. They speak the rate at which reaction approach equilibrium. Enzyme activated by ATP and other energy carriers. There are four features of enzyme, which is catalyst, which can speed a specific reaction enormously. It also can help lower the energy barrier. It also specifically recognizes the reactant and it can act repeatedly. For activation energy, for a reaction to occur, energy barrier must be surmounted. So enzyme make the energy barrier smaller. So this is the illustration of the reaction with and without the enzyme. So this show the energy needed without the enzyme. This show how much energy needed with the enzyme. Next, we move to the how catalysts work. Okay, basically hydrogen peroxide is a very toxic to the cell. So when the hydrogen peroxide enter the cavity in the catalyst is enzyme. Okay, it is a subject for reaction aided by an ion in a heme group. So what happened here, the hydrogen of the peroxide is attracted to the histidine, an amino acid projected into the cavity, and one oxygen will bind the ions. So what happened here, the binding destabilizes the peroxide bond which break and the water can be formed. In a later reaction, another hydrogen peroxide will pull the oxygen from the ion, which then will be free to act again and again. Okay, for the transition state, substrate molecules are brought together. Substrates are oriented in ways that favor reaction. Active site may shut out the water, and it also due to the induced feed model. Factors that influencing the enzyme activity are temperature, pH, salt concentration, coenzyme cofactor, and also allosteric regulators. For effect of the temperature, like a small increase of temperature can increase the molecule cohesion and also increase the rate. So high temperature may disrupt the bonds and destroy the shape of the active site. So here, let's say the higher temperature, the activity will become higher, but at certain limit of the temperature, okay, the reaction will be goes down, maybe due to the destruction bond of the enzyme. So effect of the pH, some enzymatic may react at the different optimum pH. Okay, here like enzyme A may be actively at the pH at acidic pH. When it come to neutral, okay, it start to deactivate.
Ada benda yang tak kenal. Saya boleh tolong.
Okay, some enzyme may be need enzyme helper. Enzyme helper is also known as a cofactor. Cofactor is a non-protein helper for catalytic activity. There are two types of cofactor. First, coenzyme, which is organic. It accepts electron and hydrogen ion and transfer them within the cell. Mostly it derived from the vitamin. Example of the coenzyme is NAD+, ADP, and also FAD. Second is the metal ion, which is inorganic enzyme helper, mainly in the form of ferrous ion in a cytochrome. Okay, now we move to the allosteric activation. Allosteric activation results when the binding of an effector molecule to an allosteric site cause a change in the site that makes it capable to binding substrate. Like here from the illustration, this is the enzyme. Okay, this is the active site. Okay, this is the allosteric site. So what happened here? The enzyme cannot bind with the substrate. So when the allosteric activator bind at the allosteric binding sites, so the shape of the active site will change compatible to the substrate, so the substrate can bind to the active site. So the reaction can happen. So now we move to the allosteric inhibition. Allosteric allosteric inhibition results from a change in the shape of the active site when the inhibitor binds to an allosteric site. So from here, the illustration here, this is the enzyme, this is the substrate can bind at the active site. So when the allosteric inhibitor bind at the allosteric site, so what happened here? The active site of the enzyme change the shape, okay, so the enzyme cannot bind with the substrate anymore. Okay, lastly is the feedback inhibition. First of all, the substrate can bind with the enzyme and produce the products. Okay, so this is the series of the reaction and produce the end products. This process occur repeatedly, okay, until the certain amount of the end product has been produced, okay. The product itself can inhibit the enzymatic reaction. So the whole process of the enzymatic reaction will be shut down and no more product will be produced by this reaction. So this is what we call as a feedback inhibition, when the limited or the excessive product itself can inhibit the reaction itself. For the conclusion, the cell metabolism involves many elements in order to regulate the cell organization. Cell metabolism involves in energy conversion, ATP production, enzyme mechanism, and also metabolic pathway. Okay, thank you for your attention.